I want to welcome you all. Happy 2016. This is the first of the College of Science Fridays lecture series for the 2016 year. And it's my pleasure to have Dr. Pat Gillevet, who heads the COS's MicroBiome Analysis Center, to give the first talk of the year. The microbiome, I'm sure that Pat will describe it in much greater detail. It really has, over the last
decade,
I would say decade, has become one of the central pieces of human health and biology, because as more people have realized that the interaction between the microbes and the humans is much deeper and much more symbiotic than the pathogenic interactions that people have thought about in the past. And with that, I'll let Pat explain how this intricate interaction actually has evolved over time and the kinds of findings that he's made over.
I will mention that he twisted my arm to do this first one. Okay. No. Okay, thanks. I'm going to stay in here. I'm actually colorblind. Okay, so it's easier for me just to use the mouse here. I cannot see a red pointer. All right. So I'm going to talk a little bit about this whole interaction. You are an ecosystem, is what it really comes down to. Okay. So to start with, I'll give you some concepts of what we define as the human microbiome, the Human Microbiome Project. I'll give you a little smidgen of some of the technology that has come along to allow us to actually look at this stuff. And then talk a little bit about the concept of systems biology and how to look at the nonlinear interactions of these complex dynamics. And then finally, we've been doing a whole bunch of different projects. I kind of pulled out some projects that fit into this concept of the gut-brain axis. How does the bacteria affect how you think? All right. All right. I want to introduce the gut microbiome. Okay. This is E. coli is the first bacteria that was isolated from the gut way back in 1886. It was obviously isolated by Theodore Isherek. The big question is the E. coli the most abundant bug in your gut? And the fact of the matter is, it is not. Okay? It is a facultative anaerobe, and it's the one that grows when you plate it out on a plate. So people have a very skewed view of what's in the gut. So you can think of the microbiome project starting way back in the mid-1800s. I have to mention Menshnikov. He was a microbiologist, and he got a Nobel Prize in 1908 defining phagocytosis. In fact, he used to have some pretty nasty confrontations with Pasteur, because Pasteur would not believe that a white blood cell would eat a bacteria. All right. He really was the one that really defined probiotics. All right. He thought that as far as gerontology goes, toxic putrefying bacteria made you age. And frankly, he ate kefir all his life, which is a fermented milk, and he lived to be 71 years old. And at that time, that's a fairly old person. Okay, so he had something going there. So you can think of the Human Microbiome Project as at least 100 years old. All right. So in 2005, we've been working on the microbiome project, microbiome, since about 2002. The actual microbiome project was established in 2005. And basically, we're looking at any surface that's exposed in the human body has a biofilm, has its own microbiome. And people talk about, if you take a look at the number of prokaryotic cells, there's 10 times as many prokaryotic cells in the human ecosystem than the actual eukaryotic cells. Right? And you're getting some ideas when you start talking about 10 to the 14th cells per gram. That's huge. Even in our cell cultures that we grow in the lab, they don't get up that high. All right, or rarely, anyways. All right. fundamental concept here is that the microbiome forms a biofilm. So when you talk about the Human Microbiome Project, you're looking at biofilms. We actually have other projects, environmental projects. We're still looking at lobster shell disease, and a lot of the coral diseases involve a biofilm that actually destroys the tissue. This one here, the lobsters have a biofilm on their shell. It gets distorted. It's dysbiosis is the term, and it eats away the shell. We're still doing work on this. The lobsters don't taste that bad, okay, but you can't sell them. Right? That's a problem, all right? And actually, eventually, the lobsters will die. Right, we see here, this is a mucosa, this is a villi in your gut, and this is the biofilm that's coating that mucosal barrier. It is innate, it's pretty stable, and it develops when you're a neonate, and you keep it for the rest of your life. If it gets disturbed, you get disease. You get inflammatory bowel disease, ulcerative colitis, okay, IBS, all right? This is just a blow up of it. So there's kind of two layers, and what happens here is when you develop it as a neonate, it recognizes as self. And then if you disturb it, your body recognizes it as a foreign antigen, and you get an inflammatory response. So a lot of these diseases are not infections, they're autoimmune diseases. All right. This is a blow up of the cross-section of the ileum, and these are Peyer's patches. In the human, 80% of your immune system is in the gut-associated lymphoid tissue, the GALT. Right? And if you take a look at the blow up of this Peyer's patch, this lymphoid tissue actually has connections to the interior of the lumen of the small intestine. Literally, it's talking to the bacteria. A lot of the quorums, we'll talk a little bit more about this, but a lot of the quorum-sensing molecules, molecules that actually bacteria make to talk to each other, are cyclic peptides. And you have to have a cyclase to make these peptides. Well, if you look in the human genome, you have 2 cyclases. Why would you have those? Well, you're making quorum-sensing molecules to talk to the bacteria. The mucopolysaccharides are also quorum-sensing. All right. And this is just a model that actually it illustrates the whole concept. If you have, this is the interior of the gut, you have essentially 2 layers, mucosal layers that form a biofilm. This is developed, as I said, in the neonate. You're talking to the immune system. These normal bacteria, okay? Yeah, it's hard to say what normal is because everybody is different, okay? Depending on where you grow, what you eat, okay? What you've been exposed to. And if this gets disrupted, you get bacteria that come from the lumen into this protective biofilm, and now you have immune responses. All right. The bacteria that are in the lumen change with whatever you're eating. Okay, that's pretty dynamic. This it is relatively stable. All right. So you don't want to disrupt it. Okay. There's even an issue here that if you disrupt it, you go down to Mexico and drink the wrong water, okay? You disrupt your microbiome. How does it get recolonized? And there is a theory out there right now that the appendix is a reservoir for your normal bacteria so that can recolonize the gut. I would love to do a study. I've been asking people to do this for a while, of take a look at people that go in for a colonoscopy and look at people that have their appendix or don't have their appendix and see how fast they recover from the endoscopy. Right? Can't get you to bite. I don't know. Okay. How do we analyze the microbiome? Well, as Bob will attest to you, okay, typically in the old days, okay, still not in the old days, we still do it, all right, we culture the bacteria. We can grow them in specific sugars. This is a biolog plate here. And if the bacteria grows, it produces acid, you get a color change and a dye. We can plate them out on plates and look at the morphology of the colonies, or we can use selective media. This is blood agar. Whatever's growing on here, you don't want to put your fingers in it. All right? So the paradigm out there is that less than 1% of the natural communities are culturable. Now, that's not exactly true anymore, I think, because there's been people using more dilute media and getting more things to culture from the environment. But it's very tedious. And then there was a big shift in the technology back around 2000. People started using molecular techniques to look at the bacteria, not trying to culture everything, just to go in and look at all the DNA that's there. This is the ribosomal RNA. You don't have to worry too much about the details, but the bottom line here is, if it will work, we have primers that bind to conserved regions of the ribosomal RNA and amplify across these loops which vary by species. They vary in length and in sequence. So if I have 3 different species in the sample, and I amplify it with these 2 primers, I'm going to get 3 different size amplicons. All right? PCR is polymerase chain reaction. It makes a copy of a segment of a DNA. So I'm going to get 3 different size amplicons. If I run it on our fluorescent sequencer, our workhorse that's been in the lab for 20 years, all right, you're going to get 3 different peaks. And the height of the peak is proportional to how much of that taxa was there. We call these operational taxonomic units because we don't know what they are at this point. We can distinguish them, but we don't know if it's a species or a genera or a strain. We don't know anything about it. We can't put a name on it, put it that way. All right? Then what do? All right? We would take these communities. This is actually an old project. This is a profile from the salt marsh, Al, okay, way back when. So it's fairly complex. We would clone this into a plasmid and put it to a bacteria and plate the bacteria out on a plate. We would pick these colonies and then go ahead and sequence them on the fluorescent sequencer. All right? It was tedious. If we got 200 clones, 2 runs on the sequencer for each sample, that was a lot. It was a lot of work. And it was expensive. It was like $10 a sample. $10 a clone, excuse me. So we needed to look for things that were faster and less labor, okay, and lower cost, obviously. Along came next-gen sequencing. It came around 2003 or 2004. They started coming up with new technology to do sequencing. I'm going to talk only about one of them. There's several out there, and there's some newer ones coming down the line. But this one I like to talk about because we have it in the lab now, and it's sequencing by computer. It's literally what it is. They have a semiconductor that actually looks at a pH change, and we're going to put a DNA sample in one of these tiny wells, and we're going to, each time a base goes in, it's going to change the pH, it's going to release a proton, and we're just going to look at the pH change. All right? So this is Rothberg, okay? He developed the 454 pyrosequencing, which actually looked at light, similar concept, in '99, and then he came up with the ion torrent, which looks at pH. It's kind of interesting because he developed the 454, sold it to Roche, and came along with ion torrent and put them out of business. This technology essentially, I think they stopped producing reagents this year. Okay, it's basically defunct. It lasted about 7 or 8 years, 7 years, man. Good return on investment, I think. All right. So this is actually from the Roche machine, but it's a good example to give you an idea of what we mean by emulsion PCR. All right. So typically, in a PCR, you put in your template, primers, and some enzymes. What you do here is you also put in oil, and you shake it. All right. If you shake oil and vinegar, what do you get? An emulsion. You get droplets of oil. You get a droplet around these beads, and then you run the PCR and clonally amplify the fragment onto the beads. All right. This is actually a picture of the Roche emulsion, which is big, with big beads. I can actually see them. And you can see that you have a droplet with a bead in the center. The big issue with the big change in the technology with the ion torrent is the beads are about the size of a bacteria. I was a little bit embarrassed in the class last spring. I showed them this. I said, okay, let's go look in the microscope and look at the ion torrent beads. I couldn't see anything. Kept putting in dye. They're about the size of a bacteria. Okay, about 10 microns. Okay, it's amazing. All right, and that's really why Roche machine, they can't keep up. They can't change the technology. All right, and this is just depicting this idea of an emulsion PCR. You start up with a bead that has a adapter on it. You bind one fragment, one DNA fragment from one bacterium. You amplify it, and then we basically pull out of the solution these hairy beads and sequence these. These beads then go onto the chip. This is the machine in the lab. It's extremely simple. A lot simpler than the Roche machine to run, put it that way. All right. Now this is just a blow up of your CMOS chip. So you have these little tiny wells that fit one bead. So basically they're about 10 microns across. And you basically have an ion-sensitive layer. And what the concept is, if I have these little wells, I have a bead with a fragment on it. I anneal an adapter to it, and then I flood the whole plate with A. DNA is made of A CGTs, right? So if an A goes in, it's going to release a proton. So this pH will change in that well. All right. If two of them go in, you're going to see twice as much change in the pH. And then you just wash the A off, put on a T. Wash the T off, put on a C, and just keep cycling it. So it's highly parallel. This is just a picture of the chip. The deeper the red, that is red, right? Okay. The deeper the color, okay, is the more dense the beads have been loaded, right? And this just gives you the concept. Your polymerase is coming along, making a copy of the template, and if a base goes in, you're going to see a pH change. All right? If it doesn't go in, you won't see anything. If two of them go in, you're going to see twice as much. This is Ion S5 that came out last fall, which I'm a little bit upset about. We have the PGM, which produces, Massey is getting about 4.5 million reads on a run. I'm in the third year of the lease for the machine, and I've come out with a new machine that will do 80 million wells. Okay? So machines, I haven't even paid for it, it's already outdated. Okay? So basically you can do about 80 million wells on this machine. That's 16 gigabytes. It's a genome in actually about 4 hours. 4 hours running time. It's going to take another 6 hours to analyze the data. All right? So this is just, we have the PGM. Okay, so basically you can get about 4.5 million reads on this chip. A bigger chip, you can get 6 million reads. On the new machine, it's 80 million reads. So it just gives you an idea of the throughput. This is how dramatic the technology has come. We sequenced the human genome with this machine on the left here. 96 reads at one time. That took overnight. So basically, it's about 10 million bases a year. That's if somebody is dedicated and runs that machine, and that's all they do, okay, and they bust their butt, okay, they'll put out about 10 million reads in a year, all right? 10 million bases in a year. This machine is doing 16 gigabases in an afternoon, okay? So you're basically doing 1,000 years of work in one afternoon now. That's how, and the newer technologies, you're going to start increasing this, all right? And obviously, the price is going down 1,000-fold as well per sample. All right. So the big push out there is to try to do a $2,000 genome. All right. And I give the students this little history here. In '77, they sequenced the tRNA. It's only 77 bases, and it took about 7, I should say, person years here. All right. I have to be careful. Manual sequencing. Okay, we did the human genome, all right. It was about, when I was doing manual sequencing as a postdoc, it was about 100,000 bases a year, all right. The Genome Project went from 1990 to 2001, took about 12 years, something like $2.7 billion. They figure about 50,000 man years went into this to sequence the first draft of the human genome, all right. When the 454 came out, they redid Venters in Watson's genome in 3 months for $1 million each. They've already gone down 3 orders of magnitude in price. All right? And the Ion Torrent, they're talking about one day, a couple of $1,000. All right? That's where we are. It's gotten to the point that they're setting up labs next to neonate hospitals. So when a baby's born and has a genetic disease, they'll know that afternoon what's wrong. All right. Now, they may not be able to do anything, but they're not spending weeks and weeks of biochemical tests trying to figure out what's going on. All right. That's where the technology is. Never mind looking at diagnosis and diseases and genetic diseases. Okay, systems biology, just want to make sure everybody's on the same page here. What is systems biology? We hear this all the time. What is systems biology? Okay, it's predictive modeling of complex nonlinear systems. All right. So the term first came out in the 50s when they were actually modeling action potential of the heart. Sleep actually still does this. Okay, one of the professors here over in the class now. Okay, he still does modeling of the action potential. The Russians were big on this, mainly because of Lysenko, and they couldn't do real biology. They ended up going to mathematics. All right, there's a long history there. Harold, who's here at the university, okay, came up, coined the term the biomatrix in '87. They had a big conference, him and Temple Smith. The idea of putting together the matrix of all biological knowledge and trying to, how does it all connect together? So that was really the application of, I'm going to say, the start of high throughput data collection in trying to do modeling. All right, Leroy Hood, who actually invented the fluorescence sequencer. Okay, of course he's very well off, put it that way. He's got his own institute. I think he pays for it. Okay, all right. So he established this Institute for Systems Biology, and some of the tools we're going to talk about are coming out of his group. All right. And then, of course, in 2005 we have the Human Microbiome Project. So everybody hears these issues of omics and bioinformatics. Bioinformatics is not new. It's ever since we collected any kind of biological data, you had to have some way to analyze the data. It's just the term has now come more in line with higher throughput data collection. Okay, where we like to coin the term the metabiome, which is all the different aspects of interactions in an ecosystem, a person, whatever. Okay, all the metabolites of the biochemistry. Okay, the immune system, the microbiome, how they all interact. All right. Now, of course, you have to put this in the frame of molecular evolution, especially when we're dealing with the microbiome. We're looking at how taxa are related, and we put a name on them. And we put a name on them because they usually have some functionality that we know of. We call a tiger because you know enough that it's going to eat you. We call a lamb because we know we can eat it, all right? Usually we name things for functionality. They first started naming bacteria. One produced wine, okay, the other one produced vinegar. That's why we put names on things. And we take the microbial information from these samples and we try to build a phylogenetic tree so that we can actually get some semblance of names and obviously infer functionality. All right. Put this up just to give people an idea of how variable the data is. All right. So these are actually alcoholic liver disease, healthy controls from the mucus layer and from the stool. All right. The yellow is Bacteroides, which is technically the major component of the human gut. It's an anaerobe. It doesn't grow in the plate. They never identified it initially. And what you're seeing here, I sorted it by the abundance of the bacteria. It goes from 0 in some people's sample to almost over 60%. So what's going on here? What's happening here is that this is a complex dynamic. The bacteria are going up and down. They're fluctuating. It's like a logistic equation. All right. So you happen to interrogate this sample when it was low, but that same person, if you had come, I don't know what the time frame is. I don't believe it's circadian. Okay, it's not daily, all right? But if you assayed that same person again another time, it could be actually 50%. So you're looking, and the bacteria are oscillating independent of each other, all right? The other big issue here is how to deal with this, trying to deal with this data. I only put this up here just to give you an idea that there's a lot of empty space in here. The data is non-parametric, it doesn't fit a normal curve, and it's sparse. And part of the issue here is you may have bacteria A, I may have bacteria B, and they're doing the same thing in the ecosystem. All right? So there was a big issue of moving away from just putting a name on thing to try to identify functionality. All right? So a lot of the work that we've done that really has, I think, advanced the field is not to mention the technology side, because we actually invented some of the tagging protocols, got a patent on it, is to look at the correlation networks to see what tax is correlated to what. All right? And if you have two either compounds or cytokines or bacteria, you just look at, are they correlated or not? Are they correlated? Are they oscillating the same? That's what it really comes down to. All right? And in this case, I just threw this up. I don't even remember where it came from. But anyways, these are cytokines, inflammatory cytokines. And obviously, some of them are correlated very highly with other cytokines, which makes sense. But they're also correlated with bacteria. The bacteria, again, are talking to the immune system. All right? And you can throw in here your metabolites as well. All right? All right. This is a control network that Massey did in her dissertation for bacteria in a control set of samples. The blue lines are positive correlations. The red lines are negative correlations. You can start to see which bacteria are correlated. If it's negatively correlated, that means when this guy is up, this guy is down. Or if this guy is up, this guy is down. Positive means they're both up. Okay, they're positively correlated. So you can see this network. You can think about trying to infer interactions. You can start to think about quorum sensing here, but you've got to be careful because there's a correlation. All right? In Crohn's disease, the dynamic is totally changed. It's totally different. And we can start now to looking here, see which of these bacteria may in fact be inducing the inflammatory state in Crohn's disease. And this is ulcerative colitis, another type of inflammatory. This is very different. All these bacteria here are very well tightly correlated. So it's a very different pattern. All right. You can do the same thing with metabolites. We have the technology now to go in, serum, blood, stool, okay, and look at thousands of metabolites all at once using GCMS, LCMS, NMR as well. All right. So now we're going to start looking at this whole dynamic, how it all fits together. This is an interesting paper because it emphasizes this concept of the oscillations that are going on. In this case, what they were doing, they were feeding people fiber, a prebiotic, all right? And regardless of whether they're feeding the prebiotic or not, you can see that the bacteria are oscillating. And this is over about 2.5 weeks. You see this fluctuation going on. It's not circadian. So now the real question comes in, how can we start to model these long-term fluctuations? How can we do longitudinal studies? It's not that easy, okay, in a clinical study. Much easier in mice, more expensive in mice, okay, but it's not as easy, okay? All right, so here's some examples of what's going on in this whole idea of the gut-brain axis, all right? This is a paper, it's got a nice little picture, okay, how does the bacteria influence the immune system, influences the metabolites, the circulating metabolites, and how do they affect your brain? There's a huge interaction here going on. All right, in a lot of work that we've done with VCU is looking at this concept of hepatic encephalopathy. People that with cirrhosis don't process metabolites properly. And some of the people, a certain percentage of them, get hepatic encephalopathy. They can't think straight. In fact, they can't drive. In fact, to test them, they set them down in a simulator, see how many times they drive off the road. That's what it is. All right? So the main idea here is that the ammonia produced in the gut is now not detoxified. And that crosses the blood-brain barrier, okay, and disturbs how you think. All right? What we did in this study is we took a set of people who had HE, and then we looked at the microbiome and the metabolome before we treated them, and then we treated them with rifaximin. Rifaximin is a non-absorbable antibiotic. They all got better. All right? Why? So this is the bacterial community before treatment. Bacteroides is obviously the biggest one. All right? Lactobacillus is probably the next biggest one. And then this is before, after. If I go back and forth, there's some changes, but it wasn't dramatic at all. Okay? So the microbiome itself didn't really change that much. However, if we look at the correlation network between the metabolites and the bacteria, this is before and this is after, there was a dramatic shift. So you changed the functionality of the bacteria. You didn't really change the community profile. All right? And this is actually what, if you subtract the two networks, this is what changed. Okay? This is the difference. All right? So now we want to go in and start looking at, especially these central nodes. What are these central nodes doing? What's going on there? All right? This is what's the same between the two. So you can think of this as the metabolome for cirrhosis. All right? We're bringing the concept here now of a correlation difference network. We can actually calculate the probability of whether the correlations changed, was a significant change in the correlations. So in the case one, or control, whatever, if I have a correlation between feature A or feature B, and it doesn't matter if it's metabolite, immune system, or bacteria, and it's a -1 correlation, and then in the other case, the disease state, it's 1, that was a statistical difference. That was a very, very significant difference. However, over here, C and D, this is 0.8, this is 0.2, that may not be statistically different. So the correlation difference map looks like this. So you don't draw the edge here, but you draw the edge here. All right? And you can put p-values on it. You can actually make the width of the line correlate with the p-value. Okay, so you can actually start to play some games in the visualization. All right? All right, this is the correlation difference map between the therapy, before and after. Now we actually drill down to what actually is the key changes. What we really want to do actually is to start looking at what's the dynamic of this network. This is what we're trying to do now. Try to actually calculate the dynamics of these. Look at how they oscillate. Okay, what is the trigger in a complex system like that? All right. All right. So what you're really looking at here is, with these networks, it's the linkage between the gut and the brain. All right? All right. And of course, the correlations. So here's Dilbert getting hell from the boss the boss says, every time I leave, I get a nasty email. And Wally's in the back sending the email. Okay? Correlations don't mean causation. Okay? All right? But it's knowledge discovery. It allows you to build a model and then start to bring in some, implement some mechanistic models. And actually, if you're dealing with NIH, get some money, all right? Because they won't fund knowledge discovery, all right? I put this one up too because it actually brought up some interesting issues with quorum sensing. So this is the oral metabolome, all right, looking at people that have been treated with high antiretroviral therapy. Right? People that have HIV, when they give them high heart, they actually develop candidiasis, thrush. They get a fungal infection in the mouth. All right? So what is going on here? So we looked at the bacterial biofilm, the fungal biofilm in the mouth, and the metabolites. I looked at how do these all connect. All right? And this is the correlation difference map. We can start to drill down now and see what are these. I've color-coded them by functionality, which metabolites are in peptide metabolism versus energy metabolism. If we start drilling down here, you can start to see that some of the bacteria are positively correlated with ethanolamine, which actually is a quorum sensing molecule that induces candida to form a biofilm. So instead of coming in here and giving people antifungals, maybe they should be given antibiotics, okay, to change the bacteria so they're not inducing the candidiasis, all right? And there's been a couple of other publications following up on this, all right? The other issue is, this is the PLOS paper. This paper, if you take it's kind of how hard it is to see, but there's a lot of peptides, cyclic peptides. These are all quorum sensing molecules. So the bacteria and the fungi and the human are talking to each other using these quorum sensing molecules. All right? Now, with a little I had some issues with this study. A couple of people showed up as with maize in their mouth. We're going, what the heck's going on? Well, we find out that they had been eating Doritos corn chip before they went in and had washed their mouth out, okay? So there was some arbuscular fungi in here while people were eating carrots, and they showed up in the samples too, okay? So it's a real issue with the oral microbiome and the oral microbiome is how do you actually collect these samples without cross-contaminating with food, okay? All right? All right. This is a mouse study that we just did, and I think it's in the process of being reviewed now. So this is the correlation in control mice of the bacteria and inflammatory cytokines from the cerebellum and the cortex. We have done some work with humans where we looked at MRI. Obviously, you can't dice up people's brains and look at the cytokines in people's brains, but what we're seeing here, that this is control mice, so you can see what cytokines are interacting with the bacteria. And this is cirrhotic mice. They feed the poor mice carbon tetrachloride, okay, and it fries their liver, okay? So they don't have hepatic encephalopathy, but they don't have any liver either. So you're actually looking at how did the bacteria interactions in these cytokines shift. And we're starting to come up with some ideas that, well, not just that, it's clear that it's not just metabolites that are inducing changes in the brain. It's also, you're causing an inflammatory milieu in the body, and it's that inflammation is also going to the brain and causing problems in your brain, all right? All right. Now some fun slides and just quick things that have come up lately, all right? This is the house microbiome. If you go into people's house, start swabbing bathtub knobs, bedroom floor, okay, the kitchen table, the sink, sponges in the dishwashing sponges are horrible, all right? What you see is the microbiome in the house is the same everywhere. And all the people in the house have the same microbiome. Everything in the house has the same microbiome, including the dogs, all right? And if you compare the two, okay, so what they're doing here is this is within the home and between homes, so you can see that they actually have shifted. They're different. In this slide I threw up here, this is the same individual, same house, a different individual, so they overlap, but if you go to a different home, they're different. All right. So this really is an issue now of what's going on here. Well, it's a house ecosystem is what it is, okay? And this is actually important because people that have Clostridia difficile, C. diff infections, what they're doing now is they're doing close your ears if you're squeamish, a fecal microbiome transplant, okay, from a member in the house, okay, and it cures them. They get better, all right? So basically, C. diff is always there, okay? It's an opportunistic infection. So if you go out and drink the wrong alcohol or whatever, okay, and get sick and you can get an infection. So it's actually curing. They're trying to do this for people with inflammatory bowel disease too, but we're not getting that far with the feds, okay? Because the real problem there is, what are you really transferring here? They've actually got, believe it or not, repupillate pills, okay? They spin down the bacteria, make it into a pill, and let you take a pill. Now, my opinion on that is you removed all the quorum-sensing molecules. So you're probably not going to do much with it, okay? I don't think they're having much success with it, okay? So, then...
Bacteria stimulate your cells to produce quorum molecules.
But you're upsetting that ecosystem, aren't you, okay? It's like going out to the soil here, okay, and then sterilizing, not sterilizing, but pulling all the bacteria out of the soil and then trying to get the same. It's not that same, okay? You think it might, but that was their idea. But I don't think it's going to work out that well. You need, it's like, but way back when I had fights with the clinicians at one point. They wanted, is breast milk sterile? I come from a farm, okay? The cows get mastitis if you don't milk them, okay? Why do you pasteurize cow's milk? Because it's not sterile. It's a huge ecosystem, okay? So they had this breast milk donation plant. A company came in and said, we're going to basically commercialize this breast milk stuff. And the first thing they were going to do is sterilize it. Okay, well, it just takes away all the goodies out of it. Just, okay. Anyways. I had some fights with clinicians. At one point, the clinicians were trying to tell me that your lung is sterile. Well, if you can breathe in pollen and get asthma, they said your lungs are not sterile. Okay, all right. Okay. Now, this is an interesting one that just came out. The microbiome cloud. There is a cloud of bacteria, okay, around you. These are individuals here, and this is the amount of bacteria that they find in the air around these people that are associated with normal, okay, microbiome taxa, all right? So you're shedding bacteria, all right? In fact, Schultz was correct, all right? All right? Now, it does get you, makes you thinking a little bit here, too, about, I have a new dog. What does the dog smell, okay, when they start tracking you, a bloodhound, all right? It's probably volatiles, but most of those volatiles are coming from your microbiome, all right? Now maybe it's also the bacteria as well. Okay. Okay. The last thing I'm going to show is just one thing that came out just recently: the microbiome and Alzheimer's. What they did, they found some fungal DNA circulating in people's blood in Alzheimer's patients. So they decided to take a look. It's a small sample. I think it was like nine patients. All right. And of course they're dead. Okay, because they sliced up their brains. All right. So these are the control slices. These are people with the disease. This is basically DAPI stain for DNA. They use antibodies to fungi. This is Candida showing up in the Alzheimer's patients. All right. This is tau protein, anti-tau antibodies, so it's picking up neurons, so they can put them together. So it gives a different concept to the name cheesehead, right? Okay. Now the problem with this is they're dead. These people were dead. So is this something that happened at the very end, okay, of the process, or is it actually causality of a lot of people have plaque, but they don't have Alzheimer's. They don't have dementia. Okay. So is this actually helping with that? I don't know. All right.
Sample size too. It's like 11 patients or something.
And you know how much? Well, you know, it's not a contamination issue because this is actually a significant amount of antigen there. It doesn't look like a contamination. But you know how it is with these rare microbiome samples, okay, how much problem you have with cross-contamination. Well, one of the issues we have is we always have issues with our negative controls. Well, if this is happening in our lab all the time, then no wonder we do, okay? Right? Even when we stick our hands in the hood, okay, we're blowing bacteria all over the place, okay? So now this idea of the selfish microbiome, so we have to start looking at this as an ecosystem. It's not just a gut-brain axis, it's the gut-brain, liver, immune, ecosystem axis. So I have to look at all of these together. And this is actually Conkel's paper. It was actually a nice little diagram I grabbed. So it's not just what's going on in the gut, okay, but it's also what you're being exposed. Obviously, it's going to be, the genetics is going to be involved, all right? But it's also what's going on in your house, where you grow up. There's issues now of, and this has been out for a while, we're too clean in this society. The kids used to go out and eat dirt, right? We stopped them from doing that. Well, maybe that was a mistake, all right? In fact, if you take a look at regular black soil out there, the humics, most of the bacteria that you see in your gut looks almost the same. It's hard to tell the difference. It's hard to tell the difference. Well, those last soil samples for sure are totally different, okay? But some of the stuff that we've done, you wouldn't be able to tell just from first look whether it was a gut sample or a soil sample, all right? All right, so this idea of the selfish microbiome, okay, is involved in what we eat and how it's processed, obesity. There's all kinds of studies out there about social behavior, autism. We did some work with a group down in UVA. They have autistic mice. What's an autistic mouse? I guess you put the mouse in a cage, it doesn't run over to the next mouse, it runs over and hides in a corner. Is that autism? I don't know, all right? But anyways, that's their model. They feed the grandmother mice BPA, normal amount, per kilogram, normal amounts of BPA. The first generation is fine, but the BPA causes epigenetic changes in the oocytes of the neonate. The grandkids are autistic, and now it's carried on. Now, just think about 20 years ago. We had all the women were going out drinking bottled water because they thought it was better for them, okay? Autism has gone up in this country, all right? Clearly, it's involved in higher thinking. There's all kinds of studies out there now. It's involved in development, depending on what microbes you have and what metabolites you have, okay? Involved in health and disease, heart disease, okay? So the bottom line is, you're the selfish microbe. You're just here to propagate the microbiome, okay? You're just, okay, that's it. All right. These are the groups that we've worked with, okay? Some of the bioinformatics students. This is the main core faculty in the center. Any questions? Go ahead.
So looking at it as an ecosystem, as you're doing the high throughput sequencing, are you getting any viruses? Well, I'm assuming you're getting viruses.
Okay. Well, no, because we're amplifying with specific primers for fungi or for bacteria.
Nothing for phage?
That's got to be done, okay? Okay, absolutely. Now, there are papers out there showing that the virome is highly influences the microbial community in the Chesapeake Bay and stuff. So they have to be playing an important part. All right? So yeah, that's got to be in there as well. Now we do metagenomics, okay? Which is you just take all the DNA and sequence everything. The analysis of that's a real problem. All right? So it would be better if you had more directed primer. The problem with viruses is that they vary. The sequences aren't really that conserved, okay? So it's hard to come up with primers that would, so you have to be direct to specific clades of the virus, okay? But that's got to be a big thing. And people are starting to do that. That's not good.